Group A, and today we are going to talk about Dr. Ariel Warshaw's lecture on the topic of computer simulations of biological functions. One overall motivation about Dr. Warshaw's lecture is to obtain structures and enzyme catalysts from a historical perspective. To understand what's happening at the atomic level, the electronic motions in chemical systems must be treated using the law of quantum mechanics, and a much heavier nucleus could be treated using Newton's classical equations of molecular mechanics. So using algorithms to develop a hybrid quantum mechanics molecular mechanics method can allow scientists to accurately predict the behavior of chemical systems. In order to perform some of these calculations, we need the power of advanced computers. Therefore, another overall motivations about Dr. Warshaw's lectures is using computer modeling to understand complex chemical processes, such as how to simulate long-term dynamics. Dr. Warshaw's lecture is important because it provides the foundation for many current research underlying our ability to quantitatively understand the dynamics and functions of biological molecules, as well as the structures of complex materials.
In 1950s, the structure of hemoglobin came to be solved. After that, as we can see in this picture, there was a stage where a lot of proteins could be solved. However, people still have no idea why enzyme is so powerful in catalysis. Dr. Washer wants to pick it out. A protein molecule is just like this clock. Biochemistry discovered the clock. Crystallography shows us all parts of this clock. Single molecules determines how fast the wheels rotate, which determines the speed of the reaction. In this way, we can know why enzymes can make reactions much faster. After graduation from Technium, Dr. Washoe joined the life science team to develop general Cartesian phosphate model, which can compute energy, structure, and vibrations of molecules. In 1972, Dr. Washoe used QM plus MM model to analyze this molecule. They used QM model for pi electrons and MM model for sigma bonds.
As Yingzhe has mentioned, Dr. Warshall tried to apply the combination of QM and MM approach to study enzyme catalysis. But this failed because he couldn't break any bond in simulation. So it appears that if you model molecules as bonds and springs and treat part of them quantum mechanically, we cannot understand enzyme catalysis. So the missing part is that when you break a bond, the environment stabilizes the charges of the broken bonds. As a result, we need to divide the system into two parts, the reaction region and the surrounding. We focus on the part of reaction region and put less effort in the surrounding. That is, using QM partially for the reaction region and MM for surrounding. Later, it turned out that a better approach to understand the enzyme was not to use molecular orbitals, but empirical valence bonds. It is a powerful approach because you don't need to invent quantum mechanics for the reaction. You just learn the chemistry in water and then remove water and put in the protein by changing the surrounding with EVB. So here are some applications of EVB. It's used to simulate how RAS gap complex catalysis GTP hydrolysis. In the set simulation, we can get meaningful numbers by doing free energy calculations, which are compared to actual experimental data. It's also used to study mutations of different residues. Dr. Warshall and his collaborators also solved the puzzle of how enzyme works. The reaction in water spends a lot of energy rotating the water molecules to stabilize the charges, while the reaction in protein doesn't have to, because the protein polar groups and the charges are already pointing in the correct direction.
When you have short reactions, you should run regular Newtonian dynamics on all atoms and figure out what happened in seconds and correct. Taking from the actual simulation in this model, after light come, modular moves in this exciting state and creates strains in the protein. The main point is that you did correct the prediction very early on because it was a very short reaction and because the protein only introduced constraint and we use a similar protein in photosynthesis. And then we come to the second part, what we should do if the reaction takes a very long time and have a very long system. And if we want to know what happened, we should utilize the coarse game approach, which is started with the work of Michel. And then we told the model to understand the electrostatics, and then it start to understand the large protein functions. To model very long time processes, we do not want to do it by Newtonian dynamics, and we want to do it by Brownian dynamics, like this fish feel all the forces around, and you have to find the direct friction to do the simulation. And then we find a way to do it by taking the whole protein, applying on its very long force, so it could move in short time, and then taking a small system, two-dimensional model with friction training both model to give the same speed and force, allowing us to use a simple one without force, and do it a very long time simulation. And for example, ATP, which is a model winning Nobel Prize by figuring out how it works. And the question is how we rotate this central shaft and change the structure of these unites, and lead it to chemistry. The beautiful single modular experiment that tells you something very funny. Instead of rotating 120 degrees each step, it rotates 80 degrees. Does the chemistry and continue. To understand how it happens and could we use this structure, we take the structure this direction. The change of the structure of the subunites.
What drives unidirectional working motion of moistened unactive filaments is the barrier of energy. Trying to understand how it works, lots of important information comes from the structure. When it goes forward, the barrier is much lower, comparing it going backward. Therefore, it prefers going forward and keeping the same direction. In addition, it is better to generate a model that gives it the right direction, otherwise it's quite hard to explore the mechanism of it. And this theory is verified by an experiment of doing mutation by applying different forces. Similarly, using the same approach for different questions, for example, ion channels, the flow of ions are determined by the effect of voltage activation and the external potential. So, building the electron effects model is the key to understand how these electrons work in the molecule. In addition, it is better to use Qn to simulate the electron effects model, and the rest of the system could be simulated by Nn, which is basically the spirit of multi-scale working. Complicated and very micro part of the molecule uses Qm, macro part of the molecule simulation uses Nn. It really expedites the whole calculation on the molecular system, even though it may sacrifice some precision on the simulation. With lots of benefits from the multi-scale method, it is possible to figure out what would be the next move of the virus by combining the knowledge that the virus wants to do with its chemistry. Meanwhile, we also want to combine the drug.
We have prepared 3 discussion questions for class. The first one is to briefly describe the combined quantum mechanics molecular mechanics model. The second one is how does the cross-grain modeling approach simulate the behavior of convex systems using their simplified representation. The last one is why does the protein myosin 5 on actin 5 work in one direction?